Alrighty. All right, we are streaming to Facebook and we're recording, and my screen needs to change. There we go. All right. Oh, whoops. Perfect. All right, are you seeing what you should be seeing, Roland? Yes, okay, cool. Perfect. All right, for anyone who is joining us on Facebook or who is joining our call, if you would please introduce yourself, your name and your location in the chat. I'm in Phoenix, Arizona. My name is Emma. I work with SciStarter. We'll introduce ourselves later as well, but please introduce yourselves. We're happy to have you here. And we'll give a couple of minutes before we get started anyway. Hello from New Jersey. Oh, from high school. Awesome. Great. Is it Chantal or? I apologize. I'm trying. Oh, good to have you here. Should really look into bringing some music at the beginning of these. That'd be cool. I'm not sure if that would work very well over Zoom, but for the future. Hello from Washington. I don't know where Renton is, honestly, but that's awesome. Thank you, Shirley. Ready. And then if any of you are in from Facebook, you're welcome to write into the chat the same thing, your name, say hello. We'd love to hear from you. And there's still time to actually jump onto our webinar via Zoom if you would like. And I don't know, Roland, did you by chance drop the registration link in the Facebook, or you can if you'd like? And then we'll get started in about 60 seconds or so we can stay on time. And as a reminder for anyone watching or watching from Facebook and whatnot, or for later, this is recorded. So you'll have access to this at any point on our lovely YouTube playlist. All righty. Okay. All right, well, I'll go ahead and get started then, since we are at 3 after or so. Welcome, everyone. This is SciStarter Live. Just to get us started, we're going to introduce ourselves. I am in the middle there. My name is Emma Giles. I am a program coordinator for SciStarter, and I'll be leading us through a couple of our resources today. And also on the line, we have Roland. Do you want to introduce yourself?
Yeah, hello, I'm Roland from Lebanon. I'm a quality assurance coordinator with SciStarter, and I'll be your IT support in case you needed anything today.
Yes. And Darlene is also on the line. Would you like to introduce yourself?
Hi, everyone. I'm Darlene Cavalier with Arizona State University and also the founder of SciStarter. So thank you for joining us, whether you came in through Zoom or whether you're tuning in through Facebook Live. Thank you.
Awesome. All right, before we get started, for those of you who are on Zoom with us, or if you're on Facebook, some of this applies to you as well. We have both functions. We have a chat and we also have the Q&A available. If you would like to use the chat option, that's specifically to share messages, to comment on things, anything you'd like to write in there, it's all good. If you need help with technical issues, you can send a direct message to IT support, which is Roland. It's on his name. And then we can get you the help that you need. The Q&A is specifically for if you have a question about citizen science, about SciStarter, about the featured program, which is Eterna today. And we will get to answering those as soon as we're able to. And we will have time at the end to address any that we have not gotten to if it just wasn't the time. So we'll go ahead and get started. If you do need help finding either of those things, it should be at the bottom of your Zoom window, and it's labeled as chat or Q&A. All right. Alrighty, so before we get started with anything else, we kind of want to know who we're talking to. So for our friends who are on the Zoom call with us or on Facebook, you can answer just by writing in your answer on the comment box. But we want to know who you are. Are you an aspiring scientist? Are you a student, a parent of an aspiring scientist, a teacher, an educator, a librarian, or a researcher or project leader, a bored person on the internet? That's usually my answer. Love it. Okay, great. Just waiting on a couple more. Okay, good. Yes, awesome. Okay, so we have some of you who are an aspiring scientist, 33%, 33%, excuse me, is a teacher, educator, troop leader, etc. And then the rest are librarian or part of library staff. That's awesome. Welcome. Happy to have you here. Oh, whoops. Sorry, I just hit that button again. There we go. Awesome. I myself used to be an educator, so I look at this from a perspective of an educator and a student. Have you ever been to a SciStarter live event before? So for anyone who is a multiple attendee to these, we have them every week. So hopefully we have more. Oh, yay. Awesome. A couple more of you. 10 seconds, maybe less. Depends on Roland. Awesome. All right. So one of you has been to a SciStarter live event before. That's awesome. I'm glad to have you back. Welcome back. The rest of you, I hope we turn you into regulars. We love doing these and they're every week. So you have all the opportunity to join us. And if you miss it, you can always see it online. Awesome. All right. So today we will be talking about the project Eterna, which is about RNA. We have our guest speaker, Jill Townley, with us today. She just turned her video on so you could see her. She'll introduce herself later as well, but just as a brief description, she is a Eterna player developer and Eterna game administrator and RNA designer. Started as a citizen scientist on Eterna as far as I believe last time we talked. Yes. Okay. Awesome. And we'll get to hear more about her when we get to Eterna. Awesome. Thanks, Jill. All right, so before we get started, let's get a little bit of background on what we mean by citizen science. So for anyone who is joining us who is new to the concept, new to SciStarter, new to anything in that realm, citizen science is a collaboration between scientists and those of us who are curious, concerned, and motivated to make a difference. It's how people can make an impact on issues they care about and help science. So that can take place in a myriad of different ways. You can join an app that's about climate change. You can join an app about biodiversity or different projects that deal in these things, and all of them are about making a difference and helping science. So we want to know, have any of you actually participated in a citizen science project before? Another poll will be popping up in a moment here. But I know before I joined SciStarter as a program coordinator, I had only done like 2, maybe, very few, but I knew about it. And we want to know, oh, wow, 100% of you, or at least, yeah, wow. Awesome. That's great. All of you have been participating in citizen science before. That's awesome. Well, welcome back. I'm happy that you're still in it. Oh, wait, just kidding. Okay. So to give a little bit of context for those of you who are just joining us, I saw a couple more of you join on. SciStarter is the organization that is putting on this live webinar, but also a online citizen science hub that essentially gives you access to all the different citizen science projects that exist if they are putting their projects on our site. So it's globally acclaimed as online citizen science hub, more than 1,600 projects made all over the world, organized and made searchable. So this is our search engine. You can just put in any keyword and find out what type of projects exist in that realm of space. And the first step to get involved is always to join SciStarter.org, create an account. And as a part of being a member, a person connected to SciStarter, rather having an account, means that you can also have access to our trainings. So we have a Foundations of Citizen Science training that is essentially the basics of citizen science and how to participate. It allows us to educate people on what it means to be a citizen scientist and how citizen scientists get involved. You can also earn a personalized badge with this training and with all of our trainings, because it's essentially I want to say the word timestamp, but it's personal to you. So once you've done the training, you have evidence in a digital badge that you can put onto your social media. I have it on my LinkedIn, and share it with the world so people know that you are knowledgeable in citizen science and help others understand it as well. We have other trainings as well. So if you continue on, we also have a data literacy training, which in my mind is incredibly helpful for students and teachers alike. I was a 5th grade teacher back in the day. And so I really wish I had used this during my time teaching, because it would have really helped. We also have libraries like community hubs for citizen science. So the individuals on here who are librarians, this would be helpful to you. And then teaching in higher education with citizen science is our newest one, which is helpful for seeing how we can use citizen science in higher education. And we also have an event for that specifically in SciStarter Live this month as well. All right, so today's project is Eterna, and I'm going to go through just some brief description of what this project is on SciStarter. So if you go to scistarter.org/education/Eterna-education, which Roland has just dropped into the chat, that'll lead you to the project page for Eterna. This project page tells you everything you need to know about the steps to get involved and ways to help you out. It is a SciStarter affiliate. So just to give you a little bit of background on what that means is that we have a special relationship with Eterna where we have more communication between Eterna and SciStarter, allowing us to actually track your contributions. And so we can help you get started by getting you the participation link, but then also when you go on to Eterna, we know how often how frequently you contribute to the project. And so you can see all that on your SciStarter dashboard as well. It'll say, oh, you're a frequent contributor to SciStarter, and you'll have like a link of how many contributions over time. And the only way to make that happen is because individuals who sign up through SciStarter also make sure that their email that they use on SciStarter is the same they use on Eterna. So that's the #1 step to take when you want to track your contributions. So it's not just moving onto Eterna's website and getting started. You need to make sure your account looks the same. And then only your submissions to lab puzzles will be credited to your SciStarter dashboard. So this is after you've done your tutorial. Alrighty. All right, so before we actually talk about what Eterna is, we want to know, for any of you who are educators, do any of you teach RNA in a course? Maybe? I know in 5th grade that was a little too young, but when the vaccines came out, the mRNA vaccines came out for the COVID-19 pandemic, we spent an entire day workshopping RNA because I was thinking, well, you may be 10, but this is really important and very much affecting you. Oh, that's awesome. Okay, so yes, more than one day. Yes, very briefly. No, not all. Okay, so it's pretty split between the 3. Okay, interesting. Okay, cool. All right, so some of you are familiar and some of you not as familiar, but or maybe you know more about RNA than you're supposed to teach, right? Awesome. Have any of you heard of Eterna before? This will also pop up as a poll in a moment here. I have an Eterna account. I've heard of it, but never played. I've never heard of it before. Okay. Oh, looks like it's pretty split. Some of you already have an Eterna account. That's awesome. Okay, great. And some of this will be geared towards how to use it in a classroom. So hopefully that'll be helpful as well. And for those of you who have never heard of it before, get ready because we're about to switch to Jill taking over our slides here. So I'm going to go ahead and stop my screen share. So let's talk about Eterna. Jill, let me double check if you can't screen share. Let me make sure I can make that happen. Allow. You might already be able to. Yep. Sweet. Okay. Awesome. Okay.
Great. So it's up. Okay, hi everybody, I'm Jill Townley. I started playing Eterna in 2017 and really got into it and enjoyed it. And then last year I joined the development team. And now I work with the researchers at different universities to understand their project goals and their RNA research and how Eterna can work with them. And I create the puzzles for the lab projects also. I love what I do, it's so much fun. So I'm happy to be here today and talk more about Eterna and how you can use it in the classroom. So what I'm going to do is I'm going to explain what Eterna is and how it works. And then I'm going to walk through the first tutorial to show the puzzle interface. I'll give a few ideas on how to use it in the classroom, and then I will talk about our recent projects. All right, so let's get started. First, I'm going to show you a video that's 1 minute and 30 seconds, but it gives a great overview, and you might want to play it in your classroom.
Eterna is a puzzle-solving game that reveals the mysteries of one of the most powerful molecules in biology, RNA. RNA is the companion of DNA, taking genetic information and putting it to work inside our cells by folding into complex structures. Smart RNA has the potential to transform medicine, tackling some of the most prevalent and deadly diseases on the planet, like tuberculosis, malaria, and cancer. But scientists don't fully understand its behavior. That's where Eterna comes in. Players use the 4 nucleotides that make up RNA to design a pattern that forms into a target shape. As they progress, the puzzles become more complex, and the challenges reflect the mechanics of nature. After gaining experience, players reach the lab, where their designs are manufactured and tested at Stanford University, often tens of thousands at a time. The experimental results are then returned to the players, so they can improve their designs and become scientific collaborators, joining professional scientists on the frontiers of biochemistry.
Okay, great. Eterna is a citizen science game where anyone can contribute to biomedical research by solving puzzles. Players are presented with an objective and submit designs they think will do well or give them insight on the problem being studied. We then vote on the designs, and those get tested in labs. So some of our projects, we test thousands and everything gets tested, and others we actually have to vote in because there's limited slots. The sequences are then synthesized and tested, and the data is directly returned to players who then analyze the data, learn what does and doesn't work, and hone their skills. And then we do it all over again. And we usually do several rounds of this to really learn more about RNA structure or whatever issue we're studying. Our principal investigator is Rizu Das at the Stanford School of Medicine. We're funded by NSF and NIH primarily. We were founded back in 2011. And we currently have 34 papers that have been published in major journals. Eterna uses folding engines to model how RNA can fold into different shapes. Secondary structure is predicted based on which bases in a sequence will pair according to nearest neighbor thermodynamics. And we use several different folding engines to do this. So since it can fold into different shapes, how do we know which one is the right one? Here's a short sequence as an example. Here's 3G, 3A, and 3C. And as you all know, there's 4 nucleotides, or the teachers will know there's 4 nucleotides. So G and C can't pair with A, but in this example, the 3 Gs could pair with the 3 Cs, or the 2 Gs could pair with the 2 Cs, or the 2 Gs could pair with the 2 Cs. Folding engines determine which shape is most likely by calculating free energy values. The minimum free energy shape is the most common shape. So we'll see here, this is a longer sequence that could fold into several different shapes. This one's much more open and unpaired to start with. So this is minus 4.6 kcal, then minus 7, minus 9. Then we get down to minus 12, minus 13, and minus 16. This is the lowest free energy shape, and this is what we will display in our game. But in nature, actually, RNA molecules are constantly moving a bit. Eterna puzzles take advantage of human problem-solving capabilities to solve puzzles that are computationally laborious for current computer models. Creating synthetic designs to carry out a specific function and predict how well they will perform when created is extremely difficult. But the diverse community of Eterna players represent a wide variety of ages, locations, and backgrounds, have tackled the challenge head-on and have done so with great success. The best players still remain better at designing synthetic RNA and solving secondary structures than the best computer algorithms. So that's a little explanation of how Eterna accounts works. Hope it wasn't too technical to get started. But when you go onto the Eterna website, this is what you'll see. This is our registration screen. You need to create a username, and you can't change it once you pick it. So pick the one you want and you like. And then all we collect is email and password. We don't collect any demographics, and we don't share emails with anyone. And then after you register, it'll start the tutorial series for you. And this is the first one. And now I'm going to switch over to the live demo. Okay, so here I am now on the Eterna site. And I'll go through this first tutorial with you. Here at Eterna, we study RNA, which is ribonucleic acid. Necessary for life as we know it, it serves as a messenger, builder, and a regulator. The building blocks of RNA are simple, but because of RNA folding, RNA can fold into a lot of interesting and complex shapes, sometimes very unexpected ones, which influence how they behave in biology and potentially in medicines. And that's where you come in. By playing puzzles, participating in labs, and engaging with the Eterna community, you can help us make groundbreaking scientific discoveries. This entire structure is an RNA. Each circle represents a chemical subunit known as a base. These are also known as nucleotides. Sometimes the circles connect to make base pairs. So we have 4 base pairs here in this example. The toolbar at the bottom has all the functions that are needed to operate within the game. And you can hover over to see what each one is. And the middle here is the palette where we have the 4 nucleotides. And we use these to change the nucleotides in the puzzle. And of course we have adenine, uracil, guanine, and cytosine. In the upper left are the objectives for the puzzle. Objectives that are currently unsatisfied have a red outline. And you can hover over to see the message of what needs to be satisfied. And objectives that are, once they're satisfied, they will turn into a green outline. And now we're going to do this puzzle. All this wants us to do is put in 4 of each of the other nucleotides. So we'll click. This is kind of what's confusing at first, is to figure out, well, how do I enter them? You just click here, and then it changes the adenine to a uracil. So change an A to a U. We got 4, so that was satisfied. Now I'll click on the guanine. You can also just drag to paint. It's a little quicker way when we are doing a bunch in a row. And here's a cytosine. And then once the puzzle is solved, we get all these exciting bubbles and music. And then go to the next, we go to the next one. Now I'm gonna briefly go to our home page just to show you what that looks like. You can always click on that home to get back to the home page. When you're doing the tutorials, you have limited access, I think, of what you will be able to see on the home page. Okay, it's taking a minute to come up. Maybe we won't. Oh, there it is. Let's help them see it better. Okay, this is our current projects we're doing on the pseudonauts. And you come down, there's our news. The bottom here, if you ever need to review the tutorials, they're down here at the bottom. I forgot to tell, we do have a chat. Here's our chat window where players are fairly active talking in chat, and we do try to monitor it most of the day. And then our menus are here. I would click on these arrows for the different menus. So this is player puzzles, and here's labs, and the challenges is our past projects, where you can read more about past projects. Community, we have a leaderboard, and EternaCon. We just had our EternaCon in July. You can watch videos that were presented during that. And then here about, you can see our publications, for example. Okay, and now I'm gonna switch, if I can get up to my navigation. Okay. And then able to reach my tabs.
Is it the Zoom is in the way?
Yeah, the Zoom's in the
Gotcha.
way. Usually if I just like stay still, it'll go away, but it's not going away.
I know, it does that to me all the time too. You might be able to minimize, yeah.
Minimize?
Yeah, minimize, and then you can switch tabs. Is that what you're?
No, is there a minimize? Control reactions, I can do a compact. Okay, I didn't do anything.
In the top left is your minimize, but I mean, you know that. I'm not sure.
I can't reach that either.
Ah, that's super weird. Are you trying to get back to your, oh.
I'm able to reach that tab. Oh gosh, darn
it. You can try pulling it from the top.
There we go, try that. There we go, okay. And then you can see it. Okay, perfect, thank you. So here's our document on educators using Eterna in the classroom, where I talk about groups. If you want to make a group with your students, you can have them all join that, and then you be able to have them all in one place if you want to give them instructions or know their usernames and how to register. And then we have 30 tutorials that are divided into 5 sections, and here we list all the tutorials. So you have that all in one handy place for what you might want to assign to students. And to see what students have done in Eterna, go onto their profile by clicking finding their name, clicking on it. I usually go to leaderboards to find them. It's a quick way to find them. And then cleared puzzles, it'll then pull up a list of the puzzles they have completed. Or you can click on achievements, and that shows these are the badges that you would see under achievements for the tutorials. Okay, now I'll go back to this.
Hey, Jill.
Yeah.
Where can you find the instructions for educators? Is that on the site?
I have the link.
Okay. So it's
not on the site, it's on our wiki.
Gotcha.
So it's going to be on 2 slides coming forward, and maybe you could put that in the chat when we get there, and maybe put it in the email you send everybody afterwards.
Absolutely. Thank you.
Just stop here for a second. Are there any questions so far that need to be answered?
Nothing immediate, although I do have a question. Because you have an instructions for educators, are you partnered with any universities or high schools to have this as a part of a classroom, or you might be mentioning that later?
Good question. We are not officially partnered on an ongoing basis. We did do a partnership with Prairie View A&M in 2020, where we were with a college class. We worked directly with them. And then I did a work study project with the Canadian University last winter, where they used our ribosome lab as their project that the students had to complete. And then actually at the end, one of the researchers came and watched their presentations at the end of the class. It was a pretty neat thing, but we know we are not. So we're definitely looking for ideas. We're wanting to get more into the education piece of it. What we see is quite a few high school and college classes assign the tutorials, and we see the students come into the chat and they'll ask for help, like, hey, I can't do this one puzzle. They'll get hung up on one, and they'll come into chat. And so we've become aware of different high schools and colleges that do interact with Eterna.
That's great. Awesome.
So here are my suggestions on how to use Eterna in the classroom. DNA is definitely taught in biology and chemistry and biology courses, the high school level, but RNA has been just a very quickly talked about, and it's becoming talked about more in high school biology classes now that it's become more prevalent and people know more about it. There's more to teach about it. So one thing you could do is a teacher could introduce the 4 nucleotides and talk about base pairing or however you in the classroom, and then pull up the first tutorial on Eterna and screencast that and walk through however many tutorials you want to show students how the tutorials work. And you could, a high school teacher could assign like the first 2 series of tutorials if you wanted to, or you could assign all 5. There are college courses that assign all 5. That takes about 4 hours for students to complete, that's quite a bit. And some students breeze right through and totally understand it, and other students really struggle and can take days. So it's not necessarily ideal for everyone. And so for that reason, it's a really good for extra credit projects. So if you have a list of extra credit projects for like a biology or chemistry class, you can put Eterna on there and say, do 2 of the tutorial series or 5 of the tutorial series, or you can grab other puzzles that we have and that you want to offer as an extra credit project. You can also discuss our open vaccine project. As you were just saying, Emma, it's come to the forefront now with vaccines and messenger RNA. When the epidemic started, we decided what we would do to contribute at being an RNA research lab was to study RNA degradation. So I'll talk about that more in a minute. But it highlights the benefits of connecting chemistry with RNA research and science. How is it? Apply chemistry principles to actual RNA research and citizen science in the real world. It's a way, yeah, you could present that. You could also talk about how scientists now are able to study RNA, whereas like 10 years ago, it was very hard to do hardly any RNA research, but now there's lots of new tools. And there's actually jobs in the field also, not just researchers, but there's other job opportunities. And then lastly, you could show students a codon puzzle or assign a codon puzzle. We used the codons when we were doing our open vaccine research, when we were working on the spike protein. You're all familiar with it uses a codon series. And each variant has a different codon series, slightly different is what's altering. And then we also have 3D puzzles you could bring up or assign that demonstrate how RNA folds in 3D, but that's kind of advanced. And there's the link to the document I was talking about. So I don't know if you can grab that. Unfortunately, yeah, I don't really, I'm not that good at all this to drop it in the chat myself.
No worries. I think Roland dropped it in the chat a moment ago, so we should be good there. There were a couple questions on some of the suggestions from the last slide. Would you mind going backwards by one?
Yes.
Okay. Could you describe DNA printing again? I think you might have touched on it.
No, I didn't talk about it.
Oh, you didn't. Okay.
So when we do RNA research, the RNA research is done at all the labs. They actually order the DNA, and the DNA makes the RNA. So of course, in our bodies, we all have our DNA. Every organism has DNA that's a double helix, and so it's very stable. And then it makes a copy of itself as RNA, messenger RNA, which then goes to the ribosome and tells the ribosome, hey, we need to make this protein. And it enters the ribosome, which then goes and gathers the amino acids from all over in the cell and makes the protein for each, according to the instructions in that messenger RNA. So that's what's used now for the manufacturing for my labs, I will give the list of sequences we need to the researchers, and they then place an order. There's several manufacturers now in the US. Those are actually DNA orders, and it gets, I don't know how they print it exactly, and it's obviously not on paper, but that's what they call it. But it's kind of like, it's more like 3D printing in terms of conceptually. But the manufacturers can generate the DNA sequences as ordered, and then those DNA sequences are sent to the researchers, who then are able to get the RNA made off of the DNA. Does that help?
Yeah, I think so. Yeah, that definitely helps me a little bit. In your opinion, this is a question from Chantal, thank you. Do you think that upper level high school students would be able to get to the point of actually contributing to your current project? The current project is in the one listed on that first page. I don't remember what it was called.
Correct, pseudonym, and we'll go into that next. And
oh, good, okay.
We do have high school students who are regular players and who contribute to our labs. We do, but I would estimate it's not many. So if you have a highly motivated, like I say, for an extra credit project, they can potentially look at it and figure it out and submit a solution. And we do want all the solutions. They really can't do anything wrong if they can figure it out. But it would take several hours, probably learning the tutorials and figuring it out. So it is within the realm of high school students, but it is rather advanced.
So more as an educational tool to get an understanding of RNA before being able to contribute is a bigger goal for high school?
I think so. I think it's more, we have found that it's more for explaining the RNA principles in the classroom.
Awesome, great.
I will say a couple technical things. It does require a high-speed internet because this is done on our servers. It's not done on the person's computer or phone. And it does use quite a bit of processing power because every time a mutation is made, all the thermodynamics are all being recalculated every single time. And it can use a lot of processing power. So it frequently makes it sometimes makes my fan overheat a bit for if I'm playing especially the labs, the larger puzzles too long. We mostly do web browser based. That's how we started. That's how I personally like to play it. But we do have an app now available for iOS and Android. And I loaded that pseudonaut puzzle for our lab this morning on my phone, and it actually loaded pretty quickly. I was surprised. It was quite playable. Okay, so can I move on?
Yes.
Okay.
Sorry, I was giving a thumbs up.
Oh, okay. Well, stop me if there's more questions. All right, because a little bit of background, mRNA vaccines are relatively new technology that came to light with COVID, and they were the first vaccines to the clinical trials and first to be approved and distributed. But RNA vaccines, even though they have the potential to transform immunization and it could be produced really cheaply, they have a bottleneck to their widespread usability. You all remember how the mRNA, the vaccine had to be stored at deep freeze temperatures, and then it kept frozen, and then once it was unfrozen, it had to be used within hours. So especially if eventually we'd like to use pre-filled syringes to immunize around the world, we would ideally like to be able to get refrigerator stability for vaccines. And then I'm sure many other vaccines are going to be developed using mRNA also. Like I started to say, in the epidemic, we're like, well, we're RNA researchers. What do we want to work on to contribute to this? And we decided we would work on the stability issue. So we looked at how can we design more stable RNAs to facilitate design distribution. And it's kind of interesting, this had never really been studied in depth. We generally knew that these large loops such as this, they tend to degrade quicker, whereas the more paired long helices, they hold up better, they hold up longer. And so the answer, we did several rounds, lots of different experiments, and one of the researchers developed a degradation analysis tool that we actually used in the game. And the players designed, first we designed nanoluciferase as our example molecule, a smaller molecule that we tested some things out on. And then subsequently, after all our findings, we have developed, the players did create different versions of the spike protein using that codon sequence that have subsequently been shared with researchers around the world for free, whoever wants to use them to test our hypothesis that we have a way of slowing degradation for vaccines. And I can say that the nanoluciferase in our DOS lab testing and the subsequent testing at Pfizer did show that the mRNA stayed stable at room temperature twice as long. So we were very excited to see those results. And we're done with that challenge. And we've now moved on. We've also the last 3 years been working on ribosome engineering, which has the potential for making highly customized materials for medicine also. So for synthetic materials and for medicine, it's called synthetic biology. And you all know existing ribosomes use a very defined set of amino acid building blocks. But for what researchers want to do, they're trying to figure out how to use the ribosome to create novel materials using synthetic amino acids. But the ribosomes don't process those amino acids very well. And the amino acids they're trying to use, here's normal amino acids, and they're trying to use cyclic beta amino acids. And it's a much larger molecule. And that's probably why the ribosomes aren't processing. So we actually have, researchers are using the E. coli ribosome, mostly it's the E. coli, in the laboratory and trying to get it to synthesize, instead of synthesizing a protein, to synthesize a different kind of material. And they actually are using it to synthesize a couple of different medicines at this point. Nothing on the FDA yet. Nothing has been approved by the FDA yet. I'll just go over this really quickly because again, this is a super advanced. This is a very advanced. A lot of our players didn't participate because this is a hard one to understand. But this was our last round we did, and we're hoping that through in-depth testing of specific positions in the ribosome, we can discover a ribosome that can handle those larger amino acids. And then we did that work with Northwestern University, and we hope to get that research results soon. So our current projects and future, there was the ribosome challenge that you saw on the board there on our homepage. Players are submitting thousands of short sequences likely to form a pseudoknot. And the reason we're working on this now is we have 2D folding engines that are fairly good. They're okay. but we aren't able to predict how RNA will fold in 3D, and we can't really predict pseudoknots. So I should explain what pseudoknots are or not. Probably not. I'll wait until we get to the.
That would be great. I was about to ask. I'm not sure what that is.
I'll wait till we get to my screenshot, and that'll be easier.
Okay.
And so that project is going to go on throughout 2023. That's going to be ongoing rounds and a really large project we're doing with DOS Lab at Stanford. And then we're going to be starting a new project on ASO therapeutics to treat genetic diseases with small patient populations. And then we're still doing ongoing research, ribosome engineering with several different universities, actually. So as I showed you, that's the home page. And if anyone wants to participate after doing the tutorials, they can go into this lab training, which has several puzzles that explain what a pseudoknot is and how to make one using our puzzle interface. And then you would want to enter the lab. And there's also instructions once you enter the lab. And then here is actually what the puzzle looks like. So pseudoknots, it's this design area here in the middle, the blank ones that are not locked. So these locked, this is the 3' UTR, this is the 5' UTR that has to be on there as part of our experimental process. And then when you make a hairpin loop, we put in a unique code for each one, and that's how the designs are tracked throughout the experimental process. So this is the designation code for each design. And we show that up in here. Are you able to see my cursor, Emma?
Yes, sorry. Yes, I can.
Okay, I wasn't sure if that was showing up or not. And then a pseudoknot, they're also called crossing pairs. So this is a helix here, a stem. Players call them stems. And typically would have just a series of stems. Well, in this instance, these lines here, we call them the banjo strings, what players call them. This is also a stem. And so the second stem is beginning before this first stem has actually completed. And that's more complex than what we've been studying up until this point. And I don't know, should I say anything more about pseudoknots, Emma? It's kind of hard to explain at first glance.
This might just be, can you contextualize again, what is the idea behind studying them? What's the end goal for this one?
Oh, yes, thank you. And so we're going to, we're creating thousands of pseudoknots, both we're just making them up and creating them, and we're also going to be looking at snippets from natural RNA to put those in as well. We're also putting in known pseudoknots. And ideally, we're going to generate thousands of sequences that get tested. And then the testing will show, did this sequence form a pseudoknot or not? And then we'll take, the idea is to create a very large data set based off of this testing that we will then release publicly. And different research labs can then use that data set to create new algorithms to create better folding predictions. And once researchers have better folding predictions and understand RNA, each time we understand RNA better, that helps all the different research labs who are working on potentially applications in medicine and treating diseases to understand RNA folding and be able to model it and predict it better. Okay. So we'll probably create our own algorithm too after we have the final data set, but I imagine it'll be used by several other research labs who will be creating algorithms based off of what we find or what they find. A lot of them will use machine learning to create their algorithms.
Does that mean that, so I guess, so this project in particular is being used by many different labs in the end, or the end goal would be to be able to share it with the public. So are other labs doing similar things to this project, or are they confined to just a lab space where they're just doing this? Is this a unique thing in the industry?
This is unique. I don't know of anyone else doing this. DOS Lab has developed a very good high throughput process. I'm not aware of anybody else developing a large data set of pseudoknot sequences.
That's very cool. I like it.
Yeah, and at Eterna, we're very devoted. We're devoted to open science because we think we need to all be sharing our data right away and to help each other learn more.
Right. Absolutely.
And then this is going to be my last thing I'm going to talk about. So. Our new project we're going to be starting up, it's not on the homepage, but hopefully in a month or two, we'll be starting a new project with University of Southern California, Santa Cruz. they approached us because they are going to be making antisense oligonucleotides to treat orphan diseases with small patient populations. Excuse me. Some of you may be familiar already with a couple different FDA, a couple different RNA drugs that have been approved by the FDA. So one was for the treatment of spinal muscular atrophy, and the other is for Duchenne's muscular dystrophy. And these are just short pieces of RNA, 15 to 25 nucleotides long, that will bind to the mutant RNA in the human body and help it produce the protein. So what happens in these genetically inherited diseases is it can just be one mutation in the DNA, and that then generates, like I said, the DNA makes a copy of itself as the mRNA, and that also has that mutation in it. So what these drugs do is the little pieces of RNA attach onto the messenger RNA to try to correct where that mutant, the one mutation causes the RNA to fold differently than it should. And so that little piece tries to attach to that mRNA to try to make it fold more like the original RNA. So then the original protein is produced. And I'll say that's what we're working on. The Duchenne's muscular dystrophy, it actually skips the exon. It actually forces the spliceosome to skip an exon. So actually this new class of drugs, the ASO therapeutics, they can take several different approaches to how they are treating diseases. And we're just very excited to be a part of this project. And I just can't wait to get started on it. So that's all I have for the slideshow. Here's a list of our research partners: Berkeley, Boston College in particular, and then we're going to be working with the University of California, Santa Cruz. We do quite a bit with Berkeley through the NSF Center for Genetically Encoded Materials is a conglomeration of 6 university RNA research labs, of which Berkeley and Boston College are members. So that's kind of how we're hooked in with them. Hope to do a lot more research with them in the future too. Here's our social media if you want to follow us at all. For Twitter, we have a young man who plays Eterna on Twitch a couple days a week. And he also does TikTok videos that are fun. And here's our website. And then if you have any questions on Eterna or how to use the classroom, please email me directly. And when we were on the slide talking about how to use Eterna there, I forgot to mention, we would love to hear from high school teachers on how any resources they could use from us. So if there's any resource that you can think of that you would like Eterna to provide that would be helpful for teaching RNA in the classroom, we would like to hear about that. And especially if you have an idea for should we create a module specifically just for high school education classes, we might potentially be doing that. We're looking at getting some grants and developing more education tools for the high school and college level. And we'd really love to get feedback from high school teachers on that.
Awesome. That's great. I love the whole idea of being able to educate on RNA early on in high school to be able to build the new Eterna players in college, especially. Like, it's just such a cool concept to tell high schoolers that they could be actually impacting real medicine creation within the next couple of years. I don't know, what is the production usually like between the research behind the item to the medicine? Do you know what the timeline is from this game to the medicine, or generally the idea of what the goal is maybe?
Yeah, so we haven't on Eterna created medicine yet. We did develop a new diagnostic test for tuberculosis. We developed the sequences that could be used for it, but then we never got a nonprofit organization who would then take it to the next step and make it actually into a test that could be used. So that's another project we did in the past, but we have not actually created any medicine yet. This would be the first time with this new ASO project. I do know that generally clinical research is several years. So it would first have to go to animal models and then start with a small population in a clinical setting to begin testing.
Gotcha. Okay. So from your perspective then, how did I know I mentioned before when I first introduced you, your move from being an actual player to to working with them on a more professional basis. How did you transition to that? I'm just curious what your goals were earlier, and then how this ended up being the life choice.
Well, I work part-time for Eterna now. I was so fascinated when I read about it, I think in the Washington Post and a couple places, it was a national magazine back in 2017. And I was like, wow, I can design RNA and it's going to get tested in the lab. That is so cool. So I really got into it and playing the puzzles in the labs a lot. And there's several of us players who've been around for several years, and we actually analyze the data results and we make a lot of hypotheses based off of that. And so like with this ribosome, we read research papers from other researchers that have put out, and we learn more about the ribosome and then, like what's been done so far and what could we try based on that. And we have a forum. We have a forum where we do a lot of that discussion among the players to think about different ideas of how we can do things. And we'll make spreadsheets to analyze the data, and we'll share that with the other players of what we've learned from the different labs and what. So it gets pretty involved. And then we actually had a couple players wrote a paper. They discovered a flaw in one of the in the chemical probing, one of the experimental processes used for testing how RNA folds. They found a flaw when you do a long string of A's that it no longer gets recognized by this chemical probing. And they were having a little trouble, needed a problem getting it over, little help getting it over the finish line. And so I actually rewrote that with them. And that got published 2 years ago in biochemistry. So that's really when they realized, oh, like she knows what she's doing. She can write and she understands and she can read a scientific paper. So so I've been co-author on 3 scientific papers now. So it's just super exciting. So they needed a new person to be working with the researchers. The other person has pulled back and doesn't really have the time to be doing it anymore. So they asked me to join the team, and I was thrilled to get the offer.
That's awesome.
Another neat story is we had our lead developer. He started as a player when he was in high school, and he got into more of the programming side, and worked a lot with our developers and learned all about computer science and programming. And he just graduated with his computer science degree from the University of Buffalo, and he's now our lead developer.
Oh, that's awesome. Great. Like it's a success mechanism for several people working on the project. That's so cool. Very, very cool. We have about 8 minutes to the end of the hour. So if anyone has any questions that are related to this, or how even the feedback that Jill was asking about, how to get feedback from high school, or what we would like to see in high school classrooms from Eterna to help on that realm. If anyone has suggestions or questions about what might be possible, we'd love to hear them. And if they come up later, if you suddenly realize, oh, I should have asked, Jill shared her email. You can also contact us. I'm going to go ahead and share my screen again one more time. Oh, yes. So if there is anything, you can send us anything there. Let me move to this real fast. This is our email at the very bottom here, info@scistarter.org. And before then, though, just so you are aware of other things coming up for SciStarter Live, we have on September 20th another higher education focus. This one was high school, but also with the intention of it is a tough one that kids can grow into, students can grow into. But on the 20th, we have a specific higher education and citizen science one that's geared towards college faculty. And we have a guest speaker from Budburst joining us. We also have on September 27th, International Moon Observation Night is coming up on October 1st. So we're preparing our community to be ready to observe the moon. And then on October 4th, this used to be an earlier one, but we moved it for International Moon Observation Night. But we want to talk about data literacy. So if you are interested in data literacy, again, I, as a 5th grade teacher, wanted to use our data literacy resources. And so high school, even more so, would be helpful. Any kind of student would be, and yourself as well, would be very much benefited from data literacy. And so all this will be available to you. And we have a blog post that we can send out that keeps you updated on all those. Okay. Yeah. So anything related to Eterna, and anything Jill can help us with, if you have any questions or thoughts about anything, we'd love to hear them. I'll wait another moment just in case there is anything that pops up.
I actually had a question for you, Jill. We do a lot of work with that, and also that was great. I didn't realize how much I did not know until that presentation. We at SciStarter and ASU do a lot of work with libraries, and I'm wondering if there's if you have any resources available, or what kind of tips you might have for libraries who want to engage people who are already using the library. You know, they have computer centers and they have affinity groups and others who use the library. Some might be using it for AA meetings or lifelong learning programs, gardening and computer science, job skills. I'm wondering, so I'll just be quiet and see if you have any existing resources, or maybe we can help make those connections.
We don't have any existing resources. No, we don't. And we basically would say anyone who wants to play is to come onto the website and register and start playing the tutorials, because those are rather self-contained. And but I'm open to ideas if you have ideas on what we should be doing.
We will. Sometimes there's like a community member. So I will. I think one of the things is maybe just sharing this event with the libraries that are part of our network, so they can share it with others who may be looking for programming for their different community groups that use the library. You know, because the library is a great place. They have all the computers, they have Wi-Fi and so forth. There may be an issue, and we run into this too. Sometimes the libraries won't do programs that require accounts and logins for safety reasons and others. So what do you do about that? What do you have? What do you do if like a teacher says, oh, I don't want my students to have accounts, but I'll have the account and we'll work together as a team? Is that a possibility?
That's very interesting. It's not currently, but that's the kind of thing we would love to know about for when we develop our new education modules. That would be really helpful to know. Yeah, we hadn't considered that. That's very interesting. We're going to have a meeting next week. I should start talking about some things, so I will bring that up. Do you think is that common?
Yes. Yeah, not in college, but and less so in high school. But there's so many like advanced middle school classes and very eager and ambitious parents who are now homeschool educators looking for programs to really push their kids. I would even say some of the older Girl Scouts that we work with, if they're part of a troop, sometimes it's the troop leader that will have an account, the teacher that will have an account, a parent that will have an account, and not the, you know, they're not even children, but, you know, not the learners or the students. We definitely encounter this in libraries, mostly for public safety and allowing people to remain anonymous when they're in the library. Partly because, like I said, there's groups that meet there, you know, for social service reasons, and they're anonymized.
A person can go on and play the tutorials also without starting an account as anonymous. They can actually go on and play them.
Okay.
But wouldn't then be able to go any further than the tutorials.
Yeah.
I was going to say,
yeah, lots of teachers could use that, because if they're mostly using it for high school, or if they're using it for lower grades, that wouldn't be contributing automatically because they need to learn it all first. Like those tutorials being accessible would be, or is very helpful. So
we have a new series. We used to have, oh my gosh, what was it? It was over 100 tutorials. It was really long. It was way too long. So we just hired someone to create this new series. In December, we launched it. It's much better. It's much shorter, and it gets right to the point. And we do have chat. We do monitor the chat in the game. But I know, like, Jonathan, our lead developer, when he started playing, his dad would be right there making sure nothing funny was going on. But we have an active community, and it's not, we don't monitor 24/7 in the chat, but there's generally someone around, and we do not tolerate any kind of bad language or behavior.
Yeah, I mean, I guess one other idea that we have seen in classrooms too is that whoever the facilitator is, teacher, librarian, whoever it might be, they put it up on a screen and they actually kind of crowdsource the answers together. What do you think you're seeing? So it's one person with an account, but they're getting information and insights from everybody in the room.
Yeah, that's a good way to do it. And we're also thinking of developing a virtual reality version of this that we could potentially take into high school classrooms for a day, you know, for one class, and students could play it on their headset. Oh my gosh. That would be cool, wouldn't it?
Oh yeah. Yeah. Looking at what you just presented today, I can definitely see that. Very cool.
Yeah. And that would require accounts, you know, but you could just go on and play a puzzle. And
Really neat.
I appreciate the feedback. Let me know if you have more ideas.
Absolutely. We're preparing already for Citizen Science Month, which isn't until April, but I think we're already starting to have ideas of bringing you back if you're up for it. And then we look at this almost as a test, an experiment. It gives us a chance to learn more about your project. We'll hear feedback from people who decide to take the survey that we send out, and then we can iterate a little bit on the event. And then we have a long leeway to really promote it specifically to colleges and high schools. And let's bring you back with your game sometime in April for Citizen Science Month.
Okay, yeah, we would.
Great. All right, Emma.
Great. Yeah, thank you so much, Jill, for joining us. This was incredibly insightful. I'm getting ready to go on and be like, I don't know anything, but teach me. So I'm excited. Yeah, so thank you so much for joining us. For any of you who are still around, this essentially ends our call. I'm going to end the recording in a moment here. If you do have any questions, again, please feel free to reach out. If you have any suggestions for SciStarter Live, you want to hear about a certain project, please tell us. We'd love to hear it. And yeah, please use all our resources. Let us know what we can do to make it better. We love talking to people.
Survey, Emma.
Oh gosh, yeah, thank you. Yes, we also have a survey. It should show up the second you end the call and leave the call. And it will just take you under 2 minutes, probably. A quick one just to let us know how we're doing. And that's about it. Yeah. Oh, I do have one. Is that from? Oh yeah. You're very welcome, Chantal. Hopefully I'm saying your name correctly. I'm so sorry. Yeah, happy to have you here. All right. Well, then I will go ahead and end the call. For any of us, for anyone joining from Facebook, thanks for joining us. Okay. And